You tried it.
Okay, so quick announcement, reminder before we get started with the stuff, talk more about muscle. Your exam is happening after break Wednesday, so you'll have a little time to settle in, study. Wherever we finish out today is where we'll finish content, so I'll put your practice exam up so that you have it and can look at it. We might continue some content on Monday when we return, but that stuff will just nest into your next exam. Sound good? Okay, cool. All right. So when we left off, we were talking about muscles. We were talking about how our neurons connect into our skeletal muscle and how that actually creates force. So we've looked at our somatic nervous system. So remember the somatic part of the nervous system, those are our motor fibers, our efferent nerves that go to skeletal muscle as their effector organ specifically. Okay. They release acetylcholine. We have our cholinergic receptors on the motor end plate, creating depolarization. So we walk through one by one, which we did do last time. We have those neurotransmitters coming down from our motor nerve, being released, being received by those cholinergic receptors here. So this is always going to be excitatory. So it's going to be creating an excitatory end plate potential. End plate potentials are specifically on skeletal muscle. They are always depolarizations. Depolarization leads to an action potential. So we have an action potential moving down the membrane of our muscle fiber. The action potential is then going down the T-tubule. T-tubules are in contact with the sarcoplasmic reticulum on either side. Sarcoplasmic reticulum is storing calcium. When we have that action potential come down the T-tubule, that's the signal for our sarcoplasmic reticulum to release calcium. Calcium, we saw when we peeked in at our structure of our actin and when we were looking at our cross-bridge cycle, that calcium is important. This ion sitting here in green, right? It's important because that's what causes the troponin complex sitting here to push that tropomyosin to the side, right? Otherwise, it sits over those myosin binding sites on the actin. And if it's covering over those myosin binding sites, myosin can't bind to actin, which means it can't grab on, can't use its power stroke to pull that actin towards the center of the cell, or center of the sarcomere, center of the cell. And then we can't contract our muscle. So we need this calcium in order to do our cross-bridge cycle at all. Okay. So cross-bridge cycle begins once we have that calcium in place, exposing those myosin binding sites. Right? We're using energy that we've harvested from hydrolyzing ATP. So breaking ATP into its components, ADP and a phosphate ion. We stored that energy from the bond in the shape of the myosin. Cross-bridge cycle, we use that energy in order to push the actin towards the center of our sarcomere, contracting our muscle fiber overall. Eventually, we clean up that calcium. So calcium gets actively transported back into the sarcoplasmic reticulum at the end of this process after that action potential. When that calcium detaches, that blocks the myosin binding sites again, and this is how a muscle fiber relaxes. So it's not reacting to a stimulus in order to relax, it just relaxes when the calcium gets cleaned up. So here we're zooming in, emphasizing that relationship of the calcium to the troponin complex and the tropomyosin. Okay, so I don't particularly care if you know the 3 parts of the troponin complex, although there are 3 parts, but it is important to know that when calcium binds to it, it creates this shape change and therefore the change in what our actin can do, right? So here we see at the top that troponin complex just hanging out, no calcium. Therefore, our myosin binding sites on the actin are covered over. So you would see this in a relaxed muscle, right? You'd have a long, stretched out sarcomere. When we're contracting, we're binding that calcium in, and myosin and actin bind to each other at their respective binding sites. So when we're thinking about how we turn these muscle contractions on and off, that's just my little scribbles at the bottom that I forgot about. Okay. So to terminate the contraction, relax, stop contracting, we have to get that calcium off. So for a muscle to relax, getting rid of calcium, which means that we need to get calcium both off the troponin complex, off that molecule, but more importantly, we need there to be less calcium in the cytosol overall. Right? If you have tons of calcium just swimming around inside the cell, it's just going to knock into the troponin again. So that's why we have to actively remove that calcium using our calcium ATP ACE in the sarcoplasmic reticulum, transporting that calcium out in the cytosol back into that sarcoplasmic reticulum. So we're just kind of sucking it up like a vacuum, basically. So let's take a look at this. Without an ad, please. Let's get it.
Okay, Wix for my business because it's massive scope for functionality and. So this video.
Oh, that's not what I wanted.
Looking at.
Who is that? What is that? I don't need to know about prokaryotic cells in this class.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel through hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as the sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus 2 adjacent terminal cisternae is known as a triad.
So those are like your lateral sacs.
The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
So I partially just wanted you to see that, like, as we talk a bit more about muscle, is helpful to remember that there's that 3D structure, right? So you saw that the T tubules, when we look at them in a picture, it's just one T tubule down into the muscle, but we actually have a whole network of those. We'll see that, right? Like every time we look at a sarcomere, we're also just looking at kind of 2D, but those are also 3-dimensional structures. So that's worth just kind of thinking about in your mind once you are thinking about like, how does this actually work in the body? Okay. So thinking a bit more about this contraction stuff. Muscle contraction is often described at like a bigger level with this concept of the twitch. So a twitch like is a muscle contraction. Right? So specifically, it's the contraction that you get if you just have a single action potential come down. So there's an all or nothing kind of property for a muscle fiber, just like there was for a neuron, right? Like either it gets an action potential or it doesn't. And we can talk about twitches at the individual muscle fiber level. We know that our motor units are one motor neuron splitting out to innervate multiple muscle fibers. So we can also talk about twitches from a motor unit perspective, because all of those fibers are going to be responding to the same original signal, that same original action potential from this single motor neuron. And we can also think about twitches at the whole muscle level. So like an actual named muscle twitching in response to an action potential. Okay, so think twitch, think contraction. There are phases to a twitch, so like phases of this response to the action potential, to this depolarization. So first we have a bit of a latent period. So it takes a little bit of time for that action potential to get to the muscle cell and then actually see contraction in the muscle cell. And this is partially because that excitation contraction coupling takes some time, right? So we have to have our action potential come down the motor neuron. We have to release the acetylcholine. We have to receive the acetylcholine. This has to let sodium ions in. That creates a depolarization. That depolarization or action potential has to travel out from where the nerve was, down the T tubules, has to release calcium. Cross-bridge cycle has to begin. So there's like a lot of steps, basically. So there's a latent period at the beginning of the twitch. Then there's a contraction phase. This is the time that tension is increasing. So tension, think kind of the pull that we're creating. So this is when the cross-bridge cycle is happening. Our sarcomeres are shortening, right? It's pulling on ends of the muscle. Okay. This happens about 10 to 100 milliseconds for a single twitch, basically. And then we have a relaxation phase. This takes even longer than the contraction phase. So this is as we see that tension in the muscle. So the measurable kind of like, think of it as like a measurement of force almost. Take some time for that to decrease back to zero as we are re-uptaking that calcium, right? We can't just like poof, erase it all at once. We're picking it up kind of ion by ion, takes some time. So relaxation phase takes a while. So here we can see that mapped on to a graph. So we're looking at time on our x-axis. We're looking at the tension on the y-axis. So we're thinking about this as like the force of the contraction. So during our latent period at the beginning, this is where we're waiting for that excitation, contraction stuff, excitation, contraction coupling to happen. So we're waiting ultimately to get calcium into the sarcoplasm, which is just the renamed cytoplasm, in order for the cross-bridge cycling to begin. During our contraction phase, we've got that calcium in our cell, contacting the troponin that's pushing aside the tropomyosin, uncovering those myosin binding sites on actin, allowing cross bridges to form. So that's happening during the contraction phase. And then during the relaxation phase, we're getting rid of our calcium. And it takes us longer than it did to contract in the first place. So we're a bit slower to relax. So as we're thinking about these twitches, we can also define isometric and isotonic twitches. And these are basically defined based on whether they move the endpoints of the muscle, like so the stuff it's attached to, whether they actually physically move those things or not. So think like and you're in your room or at the gym or something and you're planking, right? Like you're not moving, but you're definitely using your muscles, right? So we can have muscles contract without causing physical movement. We can use it to like maintain posture, maintain a position, or we can, you know, like lift dumbbells or something, right? And actually move a load. So this is how we're going to separate these ideas of isometric versus isotonic twitches. So when we think about, and check, I forget if I have a picture. Probably do eventually, but I'll draw one. So when we're thinking about a big named muscle, right? We have a muscle belly, like the pink part. And this is where you would see different muscle fibers with like their sarcomeres in them. All right, so this is obviously very not to scale, but I'm just going to draw a sarcomere in here. All right. Okay. So those are our contractile elements, right? In the sarcomeres, this is what actually gets smaller. Right? But we also have other parts of that muscle. So in lab, you've also been following like muscle tendons, right? So the tendons are those white shiny parts at the ends. Give us a little bone to attach to here. They're giving us a little femur, which I cannot draw a pelvis, so we're going to get a butterfly instead. But right, like we attach two bones on either side of skeletal muscle is kind of the point here. Okay, there's my tendon. All right, so those tendons at the end are part of the muscle. They're important for attaching to the bones, but they don't have sarcomeres in them. So these are our series elastic elements, are the tendons and other connective tissue attached to our muscles. Okay, so when we contract the sarcomeres, what we're contracting is that muscle belly, right? And so like that's what you feel like swelling up if you exert a muscle, right? Like it's balling up. You're feeling that muscle belly balling up. But the tendons are stretching a bit at first. They are kind of elastic. So we create a force when we contract a muscle. That's our tension. And when we think about the tension we're creating in our muscle, we usually want to know what's the force that we're working against. So we're going to have a force that opposes the contraction too, right? So like something we're trying to lift, something we're trying to push. So there's also a force at the end of the muscle, which is our load. Okay. All of that is to say, when we're defining these isometric and isotonic twitches, then we are comparing whether we're able to move that load. So we can move a load if our tension in the muscle is greater than the load that we're trying to move, right? So like basically we have to contract strong enough, we have to be strong enough to lift the load. You still contract if you don't lift it, right? You still are creating some tension, just not enough to move something. Okay. So in an isometric twitch, we have a situation where our load is greater than the tension, right? So we're saying something's heavier, basically, than your muscle is able to create force to lift it. Okay, so an isometric twitch, by definition, our load is greater than the tension. So if we measure a muscle end to end, like bone to bone, where we're attaching, you're going to see that the length of that muscle doesn't change, Because we're not contracting strongly enough to oppose the force of the load. So what we see instead is that when the sarcomeres shorten and contract, our elastic elements like the tendons and stuff kind of stretch, basically. So total length remains constant. And what's making up for the fact that our muscle fibers are getting shorter, the sarcomeres are getting shorter, is the stretchiness in the elastic elements. All right, so looking at a graph of what that looks like. All right, so we're starting out with a relaxed muscle, so we can have our length is where we have our muscle attached to this bar and attached to this force plate. All right, so this is our length L, right? And we could note that this is partially composed of our contractile elements. So in this muscle belly, this pink part, right? So this is the part that's going to contract. So contractile. And then we have our series elastic elements on the ends. So like this tendon part, that's the elastic part, which can stretch to a point. So in the contracted muscle, in this isometric muscle contraction, we haven't changed our length, right? You can see that that bar and that force plate still in the same spot. So length is the same, okay? But we have created some force in contraction, just not enough to overcome our load. And we can see that we've done something because we can see that that contractile part of the muscle has gotten shorter because the sarcomeres got shorter. But to compensate, that elastic portion has kind of stretched out a little bit. So if you were to record from this force plate what's happening, you'd see that the tension increases over time as more cross bridges are built. And then as we remove our calcium, we see some relaxation. Sorry, is there a question? Did you miss something? No. Okay, cool. That's fine. I'm not scolding you. I just wanted to make sure I'm not being confusing. Okay, cool. All right. So this is basically the picture of the twitch that we saw before, right? So we had that latent phase, we had that contraction phase, and then we had the longer relaxation phase. Okay. So this is our isometric muscle contraction. So like, this is basically what you're doing when you're planking, right? So you're not moving the endpoints, but you are using the muscle. You are creating force. You're just not moving the floor, right? So an isotonic twitch is the opposite. So this is where you actually do have enough force that you're creating, enough tension in that muscle to actually move the load. So the load is going to get lifted as the muscle, the whole thing shortens. So we're going to see contraction in the muscle belly, and those tendons, they can stretch a little, but now that our force is great enough, they are going to pull the load along from an end. So we're going to see when we graph this, that this looks different once we are able to actually move a load. Okay. So here we see a similar kind of experimental setup, right? So the relaxed part looks the same, but they've actually drawn on a load here to show you that we're going to move it. Okay, so we start with our muscle attached at 2 points, right? And then we see that in the contracted muscle, our length has now decreased to just here. So this is where it's attached, right? And our load has been lifted, right? So we had this go up a bit. So this is what happens in an isotonic muscle contraction. So this is when you actually pick something heavy up and you physically lift it. Okay. So if we are looking over time, we would note two things. So over a period of time, all have time in milliseconds. Let's think about the tension first. Okay. So if we're looking at the tension that we're measuring, so like the force that's happening here at this load end of our muscle, we'll see that we have a portion at the beginning that looks like our isometric twitch, right? So the beginning is kind of starting the same, right? And isotonic muscle contractions often are preceded by isometric muscle contractions, right? So you push on something heavy, it doesn't move, isometric. You push harder, it starts to move, isotonic. Okay. Once we hit this phase where our load is physically moving, your tension stops building and it just goes flat. So basically, once you're already moving something, you've found the right amount of force, and you don't have to like push harder once you're already got whatever it is moving, whether you're pulling it up or pulling it down. You're exerting the correct amount of force to get that thing to move. So now all that tension is instead being translated into the movement of the object, rather into just tension building in the muscle. So here we see that tension overcoming the load and then this tension flattening out. However, you are continuing to move the object even if you're not increasing how hard you're pulling or how hard you're pushing. So here we're looking at how short that muscle is getting, so how much we're shortening the muscle. So you can see for that same time period, until we get to the point where our tension flattens out, right? This is basically the point at which the load would move right there, right? When the load starts moving, that tension flattens out because now we've got the right amount of force. So that's when you're going to see the actual overall length of the muscle shortening, also reflecting that you're moving that end point. So here we can see the muscle is shortening for a while, then it's shortening less, eventually it flattens out. But it's shortening because we can move the load. We didn't have a graph of the shortening for our isometric twitch because it would just be a flat line. That muscle doesn't get shorter because that load is too strong. Its endpoints are firmly attached. We're not actually moving anything. So the muscle overall stays the same length. So in normal muscle activity, like you moving your body, sometimes you do see purely isometric contractions, right? So postural movement, bracing yourself, holding yourself in place. You use muscles for that, but you don't need to move something. But you're not really going to see purely isotonic contractions for the reason that the force needs to like build before you're up to enough force to actually move a load. So even if we don't make our load heavier and heavier and heavier, we're going to have a little bit of time where that contraction is isometric before that tension gets great enough to overcome our load. And overall, our twitch is following that isometric pathway until that tension, that force is great enough to move the load. Okay. And the isotonic bit is really where we see our muscle actively shortening. So our total length of the muscle getting shorter and that tension kind of flattening out. A trickier nuance, which we will not go into all of the physics of, but if you're actually thinking about lifting something in practice in real life, your load changes depending on the limb position, right? Like carrying something heavy close to your body versus like having your arms sticking out far out, right? That's harder in one position than the other. So it is the case that like as you move an object, the load is likely going to change because of the physics of that. So that also makes this more complicated than those two just like binary isometric versus isotonic choices. Right, that's our theoretical kind of building blocks, but real life is messy. Okay. There are some factors that affect how strongly you can contract. your muscle fibers. So you're gonna see some graded muscle contractions, so of different strengths, basically, so grade like gradient. One is how much tension any individual muscle fiber can produce. So different muscle fibers in different states are able to produce more or less tension. That's going to depend on how many of those cross bridges we can form between our actin and our myosin. If we can make more cross bridges, we can shrink that sarcomere more, pull harder, and create more force. But if you can only create a few cross bridges due to the position of the muscle, size of the sarcomere, that's going to be a weaker contraction. The other thing that we can think about if we're on the whole muscle level is how many fibers we have contracting, right? In a big named muscle. Not going to, but like theoretically in your mind, you could imagine one muscle fiber contracting, not going to do all that much. But if you contract lots of fibers at the same time, you're going to produce more tension, you're going to produce more force. Okay. You can also see differences in what happens when we have stimulation at different frequencies. So what we're thinking about is like action potentials essentially coming from our motor neuron, and we're going to see effects of the number of action potentials and how much space there is between them that affects their tension essentially. So we can see that as we have an action potential coming down our motor neuron, that's really what our stimulus is, right? So each of these arrows, think like an action potential from a motor neuron. We're going to see that for each twitch, right, for our twitch, this is tension on the y-axis, right? So this is one reaction to one stimulus. This is a second reaction to a second stimulus, and so on. You can see that as we activate a muscle like this, for a little while, we see tension increasing, right? So for a while, we have tension increasing with each twitch. So basically, think like warming up a muscle, right? So you're warming up, you're actually getting stronger a little bit each time as you're warming up that muscle. And then we hit a plateau, right? So the strongest you can twitch that muscle, eventually you hit that plateau. And this is called trepe. So this is this increase in force as you're kind of warming up the muscle with the first couple stimuli, and then hitting that plateau at your maximum force for that muscle fiber. Okay. So the basis of how action potentials kind of sum in muscles and create stronger contractions has to do with timing. So an action potential is going to happen on the order of like 2 milliseconds, but contractions can vary in length, and they can get quite a lot longer than 2 milliseconds. So we've got like 10 to 200 milliseconds for a contraction in response to a stimulus in a muscle fiber. So this means that as we have those action potentials coming in from our motor neuron triggering action potentials in our muscle fiber, our contractions can overlap and sum. So this is like different than when we had that all or none with a neuron signaling to another neuron, right? We've never had a stronger action potential. We always just had like more action potentials or fewer action potentials. When we're thinking about muscles and their contractions, we can contract stronger in response to a stronger stimulus or more stimuli. Okay. So here we just see that duration of specifically an isometric twitch. We know it's isometric because there's no plateau for that tension, which we would see in an isotonic twitch as we start to move the load. So we see our action potential. We see that latent phase while we do our excitation contraction coupling and get calcium out into, bless you, the cytoplasm of our muscle cell. We have contraction occurring for a while as we have that force building. And then we have our muscle relaxing as we're getting rid of the calcium. Okay, so those were our 3 phases to our twitch here, mapped onto our isometric twitch. Okay. And here we see some nuances in what happens if you have stimuli coming in kind of fast, basically. So action potentials repeatedly coming in from your motor neuron. So if you have just one action potential and give enough time for that muscle fiber to completely relax after twitching, after contracting, you go kind of back down to 0 with your tension. Okay. However, we're often going to see that we have multiple action potentials coming in before our muscle fiber has fully relaxed. So that's this summation here. Okay. So we can see that we had a latent phase, we had a contracting phase, we started to have a relaxing phase, but we didn't get all the way there. We didn't get rid of all the calcium, right? So the calcium is still kind of hanging out here, right? We didn't get rid of it all. We didn't go back to 0 by the time we have another action potential come in. So the next time we have an action potential come in, we release more calcium. And now when we start to relax again, we have even more calcium still hanging around because we didn't get rid of the original calcium in the first place. So this is part of how we have this warming up and this summation happening here. Okay. So we talked about that when we were talking about like summation and trepe, these different responses to the action potentials. Now we can see how tetanus forms. So like locking up the muscle and having it just be like permanently contracted, kind of rigid and stiff. We can see that that happens as we have action potentials coming in faster and faster and faster and hitting the point where our calcium really is not dropping off. And once they're fast enough, we can create this situation where we just have constant tension here. So this is some variation we can have in twitches and what they look like, depending on how many stimuli we're reacting to, how many action potentials we're reacting to, and how long there was between those action potentials. In terms of, all right, we're zooming back to our understanding of how we're generating different amounts of force. Right, so that twitch explanation was partially showing you that kind of like warming up action and how having extra calcium hanging around when we get another action potential come in means we have more and more calcium, and we saw that tension going up. Another thing that can affect the force generated is the diameter of our muscle fiber. Okay, so when we've been looking at our thick and thin filaments and our sarcomeres, right? We've been looking at like a very schematic kind of drawing. Okay. So we've been looking at something like this. Whatever. Okay, but this is happening in 3 dimensions. So if we were to cut a muscle kind of through its body, right, cut it like this, there are actually going to be kind of different numbers of thick and thin filaments. So it's not just all happening in a plane, right? So you'd see actin here, actin here, actin here, actin here, actin here, actin here, right? And our thick fibers are also scattered. Okay. We can have them alternating like we did in the sarcomeres, but basically this is happening at a scale and in 3 dimensions, not just in these individual like 2D views of our sarcomeres that we've been looking at. Okay, so this matters because the more cross bridges we can form, the more force we'll get. And so the more sarcomeres we have like sitting next to each other, basically, the more force we can get, right? So imagine this is a sarcomere, imagine this is another sarcomere sitting next to it in 3D space, and so on, like that within our muscle fiber. Okay. So if we can generate more cross bridges, either by starting out with an ideal amount of overlap here between our myosin heads and the actin, or by just plain having more sarcomeres next to each other in parallel, we're going to be able to create stronger force when we have calcium and start that cross bridge cycling. Okay. The reason the fiber diameter matters is because our sarcomeres just like are a size, the number of thick and thin filaments in an area is going to stay constant. But if you end up with a thicker muscle fiber, right? Like if you just make this thicker in diameter, you can stack more next to each other. Okay. So if you have a larger diameter, right? This is our diameter. It's the length kind of across that cut edge. Right. If you make it wider, you can put more sarcomeres in parallel. All right. So you could add more and more and more as this fiber gets wider. Okay. So if we have a wider fiber diameter, larger fiber, we're going to be able to generate more force because we have more sarcomeres and therefore more cross bridges. The length of the fiber at the onset of contraction also changes the force. And this goes back to still that fact that the more cross bridges we generate, the more cross bridge cycling we do, the more force we can make. Okay. So muscle fibers and sarcomeres have an optimal length. And that optimal length is going to be when you can develop the most tension. So form the most cross bridges, make the strongest contraction. And this is when we have our optimal amount of overlap between the thick and thin filaments. Fortunately, you don't have to like worry about this most of the time. Your muscles are pretty much positioned normally in your body at their optimal length. But we'll take a look at like what exactly that means about like the right amount of overlap, because I think it's going to make sense with the picture. Do I have a picture? Okay, the picture is at the end there. Okay. So while we're looking at that picture at the end, we're going to think about what would make something a bad length. So non-optimal length. If you have less than optimum length to your muscle fiber, you're not going to have enough overlap between actin and myosin to create a good amount of cross bridges. And if you have a less than optimum length, you're going to end up with like too much overlap and not enough room to move. Okay. We talked about the causes of the summation and tetanus, but it's just the amount of calcium basically. So if we have more calcium, greater calcium in the cytosol, don't reuptake enough of it, then you have tons and tons of calcium. Muscle contracts more strongly and more strongly and more strongly as we have more cross bridges form. Eventually, all of the troponin has calcium bound to it, which is when we see that tetanus kind of happening, like everything's at its strongest contraction, everything has calcium bound to it. We're not relaxing, we're not unbinding the calcium, everything's kind of maxed out there. It's all about the calcium. Okay, so let's look at these pictures because it's worth a little bit of explaining. Okay. So at your optimal length, we're talking about is those cross bridges have to form at the myosin heads, because those are what grab the actin with the binding sites, right? So this is where we have those cross bridges forming. Okay. Right. And this is a good amount of overlap, right? Like all of those heads are right next to actin, so they can grab it. You don't have any heads that like have no actin next to them to grab onto. And you've got some room to move here, right? So like you actually physically have space where you can shorten this muscle. Okay. As you get too long, right? Like if you're stretched out too far, you're in a really weak position. Part of what is happening is that you have heads on myosin that just aren't close enough to any actin to grab it in the first place. Right, so these heads, doesn't matter how much calcium there is until they're next to an actin with its myosin binding site, they can't hold on to anything. So no cross-bridge cycling is going to happen because they're just not close enough to that thin filament to pull on it. So that's the problem with too long a length, right? So this is why it's more optimal here, is that we have actin for all of those heads on the myosin to grab onto. If you get too short, you have a different problem. Okay. So as you get too short, all right, we have actin next to the myosin heads, right? This isn't a problem. We've got something to grab onto. But now our thin filaments are starting to bump into each other, right? Like you just don't have the space to pull. There's nowhere for this stuff to go to, right? Your I band is basically gone. You have the thin filaments from either side coming bumping into each other, right? So it doesn't really matter how much calcium you get in this situation because there's nowhere to go. You kind of can't make the sarcomere any shorter because you're running out of space. So this is what we mean when we say there's an optimal length to the muscle. That's when we have full overlap between the thick and thin filaments in those parts with the binding sites, but we still have room for that thin filament to move towards the center without bumping into other stuff. Okay. Final thing, we really will finish out this packet. Okay. So big end takeaway at the whole muscle level, if you have more muscle fibers contract, greater force. Muscle fibers get recruited, so used in a specific order. So if your motor neurons come in, they're telling your muscle fiber to contract. First, it's going to grab like one little section of muscle fibers, then it's going to grab a larger section and a larger section until you're generating the appropriate amount of force. So you're going to increase overall muscle tension for the situation. And when I say little groups of muscle fibers, each of those groups is a motor unit. So here we see a motor unit X and Y. So here we see 1, 2, 3, 4, 5 muscle fibers corresponding to this motor unit X, right? 7 fibers for motor unit Y. So first, when X fires, we'll contract those 5 fibers. If Y were to fire on its own, we'd contract 7 fibers. When we contract them together, we get all 12. More fibers we're contracting, more strength. So more fibers contract, greater tension. When we activate a motor neuron, it activates all the muscle fibers that are attached to it. That's what comprises a single motor unit. If it's a smaller motor unit, so fewer muscle fibers, less tension, bigger motor unit, more muscle fibers, more tension. So for delicate movements, you want to just activate the small motor units. For strength, you want to get large motor units with lots and lots of muscle tension, force generating ability. That's just repeating that, right? So this is what's up with the fiber diameter. In those small motor units, you'll also expect those individual muscle fibers to be thinner, smaller diameter for weak movements, larger diameter for those stronger movements, more sarcomeres in parallel, creating more force. And you're going to recruit these motor units in an order. So you're going to recruit small ones, then medium ones, then large ones. So we really are going to recruit first that 5 fiber motor unit, then that 7 fiber motor unit, and so on. So large units get recruited last because they take a bit longer to depolarize, just because they're bigger, right? You need that depolarization to spread more distance. You need more sodium to actually depolarize the cell. And so this is what that whole thing looks like, right? So in order, First, we generate some force with our small little delicate motor unit. Second, we got a bit of a larger motor unit tacking on. We want a really strong movement, got a big load to move. Tack on a bigger one, increasing force, increasing force. All right, that's it. Have a great break. I'll see you when you get back. Monday we'll do a little review and a little moving forward, but none of that new content will be on your exam so that you can be studying over break and have all the stuff that you need to know.